Everyone ready to start? Stretch your legs, grab your coffee. Good. So, last week Monday, we talked about sequence alignment and a couple different algorithms that we used to do so. Today I'd like to continue with that, so pick up where we left off with SciBlast and delve a bit deeper into what it is that makes SciBlast work. So this is very tightly related to protein structures, which is the main topic of today's lecture. So quick recap of what we spoke of last week. Can someone remind me what exactly is sequence alignment? Yes. Okay, very good. So we are comparing two sequences, and you already dove into the second question as well. Why do we do it? And this is related to homology, indeed. So what does it mean if two sequences are homologous? They are evolutionarily related, and in turn this means that if we know something about the one sequence, we can infer some of that knowledge about the second sequence, right? Because structure, function, and sequence are all evolutionarily conserved. Okay, then we discussed briefly a couple of different algorithms. Needleman-Wunsch for global alignment, Smith-Waterman for local alignment, BLAST, the basic local alignment search tool, and SciBlast, the same thing but now iterated. Okay. What does local alignment give us that global alignment did not? In other words, what is the added value or the benefit of only looking at part of sequences which align, rather than making the whole set of sequences align? Yes?
You can introduce gaps, and they will align better.
No, that's not specific to local alignment. So the answer was that we can introduce gaps. But we can do so in a global alignment as well. In fact, if you have two sequences of different length, you must introduce gaps in one of them, in the shorter one, in a global alignment. But there was something else that local alignment lets us do that global alignment does not. You, sir.
The length of the sequences are wildly different. Because if you use the global alignment, then you get huge penalties for stuff.
Very good. So local alignment allows us to zoom in on smaller bits of sequence that might have been evolutionarily conserved independent of the rest of the sequence that it is a part of. Right? And the example that we gave in previous lecture was the immunoglobulin domain, which has been copied a couple of times, conserved across species, but is now found in different genes more than once. Sometimes it is connected to different domains. Especially the people of you that are working with PFAM for their project work must have seen a couple of examples of this by now. But yes, very importantly, local alignment adds something to global alignment, and that is that we can now look at biologically sensible units rather than what we think is the whole sequence. Okay, what does BLAST give us that Smith-Waterman alignment did not? Lacking volunteers, I will appoint one. Anyone dare to hazard a guess as to what BLAST adds that Smith-Waterman does not?
E-value, which means how likely it would occur if you have a random match inside the database and you infer.
Very good. So, BLAST gives us an e-value. Why is this e-value important? You already mentioned it in your answer. You discussed the database. Is it feasible to perform Smith-Waterman alignment against an entire database? I hear a no. Any takers for yes? No, you were absolutely correct. Depending on the size of your database, of course, Smith-Waterman alignment can very quickly get very expensive if you have to do it 1,000,000 times for a single query. So BLAST, the basic local alignment search tool, is a heuristic method to do more or less this thing, but in a clever way so that you get results much faster. And then the last step that we only briefly introduced last week is PSI-BLAST, position-specific iterated blast. Both of these last algorithms are very critical for your project, so you have to know how these work. What does PSI-BLAST consider that BLAST did not? Alright, so I'm hearing the position. Okay, but it does something with those positions. Specifically, what information does PSI-BLAST use?
How often amino acids found in a specific position?
Very good. Did everyone catch that? Okay, I'll just repeat it for the back of the room. The answer given is that PSI-BLAST counts how often an amino acid is found in a specific position. That's where the name comes from, right? Position-specific iterated blast. And then what it does with this information is it simply counts and then it constructs a profile of what amino acids are allowed in certain positions. Okay, and this works because we know from biology that the important bits are conserved and thus will be in the same spot more often than not. Okay, so we will briefly continue with where we left off with PSI-BLAST last week. We will take a closer look at what exactly these position-specific scoring matrices contain. We will have a quick discussion of the do's and don'ts and what you can and cannot do with PSI-BLAST. And then we'll delve into the 4 different levels of protein structure and what we might predict about those computationally. So, you've all run BLAST now for your projects. If you do this on the command line, you get this neat little tab-separated table on your standard out. If you do this on the web server, you might get a graph very much like this. Right, so we see here that the query itself is 4,000 positions long, probably a nucleotide sequence then, because 4,000 amino acids is very, very long for a protein. And we can see here that we have a couple of hits that span the entire length of our query, and then there is regions within this query that have a whole bunch of hits only mapping to this region. So at a glance, can someone tell me which of these positions are better conserved than others? Yes. Sorry, one more time? Very good. What tells you this? Okay, but it's not just a score that tells you whether something is conserved, right?
Yeah, you can see that's like straight on.
Exactly. So there is pieces of sequence that are simply completely missing in any of the other hits. So apparently, these bits of sequence were not critical to maintaining the function that the query carries out, whereas these bits obviously are. Or at least, I say obviously, but this is what we derive from this result. Okay, another way of visualizing this is the same figure that we showed last week. We have here now for a single position, or actually for 90 single positions in our query, what is aligned against it. So these are a whole bunch of the high scoring segment pairs aligned against the positions, and you see again in the colored columns where conservation occurs along your query. Different way of visualizing this is in the PSSM, the position specific scoring matrix. So for each position in your query, we can indicate how likely it is a certain amino acid occurs. Now these are weird numbers, right? These are not counts or probabilities. These are actually log transformed frequencies. Won't bother you with the equation. I will show a better example of this in one slide. Okay, so the trick that PSI-BLAST performs that BLAST did not is that it uses a profile instead of a single sequence to find more hits in your database. Right, so the very first iteration of PSI-BLAST is the very same as a normal BLAST query, but then instead of calling it a day and going home with these results, what PSI-BLAST does is it constructs a profile for your query given the best hits of its first iteration, and then with that profile, that becomes the query for the second iteration. If you use this profile, you can search against the database again, and what you're doing then is instead of aligning a sequence, you're aligning the PSSM to your sequences, right? The sequences in your database. And then in each position of the sequence that you find a hit for, you get a new count of how often you find the amino acid in a certain position. And this then becomes your profile for the next iteration. A question?
When you say Cyclops uses the best hits, how do you determine how many hits to use? What proportion could number?
Right. So the default parameterization of PsyBlast does not restrict the amount of sequences you include in your profile to a specific number, but rather it sets a threshold on the e-value of your hits. And all the hits that you find below that e-value threshold, also called the inclusion threshold, are then included in your PSSM for the next iteration.
Is that often different to the e-value threshold used for like a putative formula?
Typically, yes. We'll get back to that in a couple slides. Right. So, once you've run your first iteration of BLAST, you've gathered a couple of hits, you select the ones with the lowest e-values, and from this, you create a PSSM. This PSSM looks something like this. Now, this is a lot to take in, so I'll go through it slowly. You have in the rows here the different amino acids of your query sequence. And in the columns, you have all the amino acids in the alphabet, or in the amino acid alphabet at least. Now, what you can see in the gray cells with the bold white text is self-hits. This PSSM will replace your substitution table, but it will do so in a way that you don't have to use it anymore regardless. You can just look up for position 1, if you align an A, what does this do to your alignment score, the bit score? Well, an A would find a penalty of 3, whereas a match with itself, another methionine, you would end up with a bonus of 10, and nothing else you can do in this row will give it a positive score. What does that tell you about the conservation of the methionine in position 1 of this query sequence? Sorry, a bit louder, please. It's very conserved, exactly. All right. Now we compare this, for example, to position 20, where we have a whole bunch of roughly 0 values. What does that tell us about the conservation? It's not very conserved. In fact, we see so much variance at this specific position that even a self-match is barely a bonus. And you'd be better off aligning a G there instead. That's weird, right? That means that in all of our hits, we see a G there more often than we see the D. Could you have known this if you had only searched with the sequence itself and not have gone to a profile-based method? That what your query contains is a D, but what most of its homologs have there is a G. No, of course not, because you only have your query. So this is a very concrete example of the added value of using a profile, right? Okay. Now one last thing I want to point out here is that the intensity of the color in the query column indicates how conserved this position is. Right, so you can just at a glance see that this one is very conserved, this one is very conserved, this one is not very conserved, and the sort of middle green ones are in between. Okay, so this is what a PSSM actually looks like. This is what PsyBlast uses under the hood. It's good to be aware of this. Right, so again, PsyBlast uses iteration. So you start with a simple BLAST query against your database, you find hits, you threshold them. They're called significant hits there, but it's basically every hit that falls within your inclusion threshold. You construct a PSSM, And this becomes your query for the next iteration. Yes?
Yes, I have a question about the profile. Is it a profile with just one number for each position, or is it gives like maybe 3 options which have the highest scores, or just with X?
So if I understand your question correctly, it's about whether the profile takes into account the e-values of the hits that it finds it includes. Is that
No,
your
like,
question, or
how does the profile look like? Is it just
like this?
is it just one string, or like for position 1? Well, no, it's not a good example. For position 20, you have a few that might be in that place, which is observed. Gives it like all the options, or is it just choose one? I mean, as in what would be in that position in the profile.
Okay, so the profile is this entire matrix. It's not choosing anything, it's simply counting. Right? Again, we have the query in the rows. So for every position in your query, you find the hits, and for every position that is aligned against one of your query positions, you count. I've seen this amino acid 20 times in position 1. I've seen the other amino acid 1 time in position 1. I've seen this amino acid never in position 1. And that then becomes the input for this normalization, this frequency checking. You compare this with the substitution matrix originally, and then you calculate the log odds, and that comes up with this entire table. Right? So this also means that if you have, say, a leucine, the L, in, 7 different positions, or 8 even, not each of those positions has the leucine conserved equally often. And it is very possible that conserving the leucine in one position is more important to your alignment than conserving it in the other. For example, these two have scores of 6, whereas this one has a score of 2 for aligning another leucine against you in your database sequence. Does this clarify your question? Any other questions before we move on? Yes?
Why are there no gaps in this matrix?
Right. I'm actually going to save that question and give it back to the audience in 2 slides. Right, so this is the workflow of a PsyBlast query. You've seen this before, I've seen it again, never forget it. Why are there no gaps in your PSSM? So, given a protein sequence of 100 amino acids, we run BLAST, obtain 25 hits to sequences ranging from 50 to 250 amino acids themselves. Now, for the sake of simplicity, let's assume that every single position of your original 100 amino acid query is covered at least once by your 25 hits. Out of this, what are the dimensions of this PSSM we now construct? Close, but not quite. Sorry, what was that?
100 by 250.
Also close, but still not correct. So the 100 rows, that bit is correct. But what was in the columns again? You, sir.
20 by 100.
Very good. So which is the rows and which is the columns? Yes?
The 20 is the.
Alright, so 20 abroad and 100 long. Correct. Does everyone understand why?
Yeah, because 20 is the amino acid, you only have 10 amino acid, and the row is your favorite.
Very good. So every position in our 100 amino acid query has a row to itself. And then for every row, we count, or we score, how often each of the 20 amino acids occurs. So for a 100 amino acid query, we must end up with 100 by 20 PSSM. Now, if we would start the BLAST run using one of the 25 hits instead of the original query, would we end up with the same PSSM? See some people shaking no, see some people nodding yes. Anyone care to explain their head bobbing? So, while you would expect roughly the same actual position scores for the aligned positions, the hits themselves have a different length. So the number of rows in our PSSM, if we start with one of the hit sequences, must be different than the query sequence. Now, back to your question. Why are there no gaps in any of our PSSMs?
They just aren't counted on the scoring metrics. So there are gaps in the queries or in the side blasts, but they just don't get counted for amino acid.
Very good. So if I may paraphrase that slightly,
Sure.
there is never any gaps in your query. The query itself is just a sequence. Gaps are introduced by alignment. But because these PSSMs are made through master-slave alignment, there are no gaps in your rows. Now what you can do is count the gaps, or actually the number of hits that do not have a residue aligned against your query position. So you can add a 21st column to score how many gaps there are, but that's for the next course. Let's not go into too much detail there right now. So, concrete answer to your question, because your query doesn't have gaps. Okay, now, final question before we, or sort of a quick scenario, before we stop talking about sideblasts for the rest of the day. We have a protein sequence, but we do not know the structure of this protein. Now we want to find a sequence with a known structure that is similar enough to our query protein that we might infer some information about the structure. So, obvious place to do this is the protein data bank, the PDB. Those people working with SCOP will very well know exactly what information is in there, and I cannot imagine that it has not been mentioned before in any of the other lectures. 70,000 protein sequences and their corresponding structures. We run BLAST against these 70,000 sequences, and we find no hits. Would it have helped to run PsyBlast instead? And why?
It wouldn't, because the first iteration of PsyBlast is identical to BLAST, so you end up with 0 hits.
Right. This always trips up a lot of students. Your answer is correct, and all of you need to remember it. Right? If you do not find a hit with BLAST, then you will not find a hit with PsyBlast, because the first iteration is BLAST. What could we have done differently?
Raise the e-value thresholds.
Possible, yes, but that risks including a lot of false positives. Is there more information that we could find somewhere that would have solved this issue with no queries from the sequence alone?
You can run blanks on a different database to get a profile that you can run on PDB.
Did everyone catch that? There's a question on this in your project reports. So the suggestion is to run BLAST on a different database, gather your profile information about what positions are conserved or homologous from there, and then proceed to use this newly constructed profile to research the PDB database. Suggestions for which database you might use for such a profile query? Where would you want to look for finding similar sequences? Because this PDB database is fairly constricted, right? We need something bigger. We need more information about the evolutionary neighborhood of our query sequence. Where might we find such a thing? Uniprot's a very good start, yes. Any other suggestions?
NR30.
Very good. Everyone know what NR30 is? This is the non-redundant database. So every sequence we have ever measured is added to NR, and then NR30 is that same set of sequences filtered down, so that every sequence is at least 30% unique. If you are less than 30% unique relative to the other sequences in the set, you don't make the cut. So So NR30 is a smaller set of non-redundant sequences that is still searchable. We have NR30 somewhere on the PCOM server, so you can actually use this for queries during your project as well. Ask one of your TAs to help you figure this out towards the end of your projects. Right, so this is exactly how you could solve the problem. You start your BLAST query against a bigger sequence database. From there you construct your PSSM, and then with this PSSM you go back to your PDB database, and that is where you can now fairly look for homologous sequences. Okay, we've just been through this. We have briefly discussed what to consider when setting an e-value threshold for inclusion in the PSSM as well. Just very briefly, I'd like to mention something called profile wonder. An earlier suggestion to find more homologs was to increase the accepted threshold. Problem with this is that you add a lot of false positives, right? Because a lot of the sequences that aren't actually homologous will now start falling within your inclusion threshold. If this happens not one iteration but several in a row, what will end up happening is that before long you've added your entire database to the profile, or at least a very substantial portion of sequences that aren't actually homologous to your query. The other end of the spectrum is equally bad. If your inclusion threshold is too stringent, you will not add any sequences at all, or you will not add enough information about the homologous sequences to find the more distant ones. So this is a bit of a balancing act, and there is no one single right way to do this. What I would recommend is that you play around with this a little in your own project database, and you get a feel for what is the consequence of raising or lowering this inclusion threshold too far or too little. Right, just quick slide to drive home the point that this really works. SciBlast for distant homology searches on protein sequences very strongly outperforms BLAST, as you can see from the barely visible yellow line here versus the purple line slightly above it. More sophisticated methods that perform profile matching instead of profile sequence matching perform even better. If you want to know more about this, follow the course Algorithms and Sequence Analysis next period. Jaap and Kim Scholander are experts in this topic, and they will very happily tell you more about it. Okay, so why does profile sequence searching work? They help us to find more distant homologs because they take into account the entropy or the conservation of specific residues in specific positions. What do these represent? It's famous Spanish from the Bill Clinton era of American politics. It used to say, is the economy stupid? But these profiles represent structure. And that is what we will be talking about the remainder of this lecture. Skipping this. Yes. So, protein structure. As promised, there are 4 different levels to this. The primary structure, or simply the amino acid sequence of a protein. The secondary structure is how these amino acids collapse into local substructures like alpha helices, beta sheets, or coils. This is related to how the backbone of an amino acid chain is formed and the hydrogen bonds that they form between each other. And there is the tertiary structure, which is the global conformation of a protein molecule. This is driven by hydrophobic collapse and further alignment of the secondary structures. And then finally there's a quaternary structure, which is the complex that a protein forms either with other proteins, with other molecules, with smaller ligands. So everything that a protein interacts with, and ultimately the thing that gives a protein its function. So these words that I've pasted behind the primary, secondary, tertiary, quaternary structures are concepts related to that level, and you should know these well by the end of this course. And then towards the end we will talk a bit again about what we can predict about these different levels. So, as we've just mentioned, levels of protein structure, the primary one is simply your amino acid sequence. So this goes from the N to the C terminus, always starts with a methionine, post-translational processing notwithstanding. And then my cat is eating fish and me eat water? No. There is contests for finding the longest words in protein sequences. And Anton, correct me if I'm wrong, but I think champagne bottle was found not too long ago.
I have not been keeping up with this. I got as far as champagne.
Right. So there are synthetic biologists who, as a hobby, insert their signatures into the proteins that they construct. Primary sequence, amino acids. Secondary structure. Can anyone explain to me what this notation indicates? Beta sheets and alpha helices. So which one is which? Okay. What about these black lines in between? Loops, loops, coils. Yes. Less structured bits of protein. Okay. We'll get to what is a beta sheet or an alpha helix and how do they form in a couple slides. Okay, so now we have a series of amino acids, and we know that these like to be in a beta sheet or an alpha helix, and then these things want to fold again in space. So this becomes a 3D conformation that is known as the tertiary structure. It's also known as the topology in SCOP, or architecture in CATH. Tertiary structure, tertiary means 3, so it's the 3-dimensional conformation of all your atoms in space. And then finally, the quaternary structure is the complex level, as we've briefly mentioned. Proteins don't act in isolation, but rather they give biological function by interacting with their environment, which can be other proteins, can be other molecules, can be anything, but they interact with stuff. And once they are interacting, they form a complex, and this complex is known as the quaternary structure. Okay. Does everyone recognize the Coulomb Table? For the scarce few who do not, pay attention during the biology refresher course. This will surely be covered in depth. The idea is that the Coulomb Table tells you how to translate a gene sequence of nucleotides into an amino acid, or into a series of amino acids, your proteins. Right? So this works from the gene level on the DNA, where you have transcription and splicing into mature messenger RNA, and this is parsed by your ribosomes in the process called translation. So every 3 subsequent nucleotides get switched out for the encoded amino acid. So the primary structure, or the primary, the amino acid sequence of a protein and its gene sequence are very closely related. There were variants of BLAST that use this information, but we won't go into too much detail there. Okay, so DNA becomes RNA, becomes an amino acid sequence, and all the way down the road of all the protein. This primary structure has an identical backbone for every single residue. So every amino acid has the same sort of general layout. This is the slide that shows that best. So you have this backbone consisting of a nitrogen and 2 carbon atoms, and this connects to the same bit of backbone of the next amino acid. What is different between the amino acid is the residue. So this is the atoms drawn in blue here. You have positively charged amino acids, you have negatively charged amino acids, you have different polar amino acids, and then you have the hydrophobes, which do not like water at all. You can see here that this is the way in which the backbone connects. So we have here a nitrogen, a carbon, and this connects to the next amino acids, nitrogen, carbon, carbon. Nitrogen, carbon, carbon, nitrogen, carbon, carbon. Right, so this backbone is just a repeating set of 3 atoms that has 1 residue stuck onto the side of it. Now, in an actual protein, the residue that sticks off to the side can either be facing the same direction as the previous amino acids, in which case they are cislocated, or they can flip opposite directions, in which case they are translocated. This is important for the way secondary structures form. There's a couple of special amino acids. First one is cysteine. And we briefly mentioned why cysteine is so bizarre, and that is that it forms covalent bonds with other cysteines, the disulfide bridge. It's critical for the structure of many proteins, and you will find it highly conserved in most cases where it occurs. The formation of this bond is a post-translational modification, and as we said, it's covalent, so it's far more stable than most of the other interacting forces that govern the shape of a protein. Right. Another weird one is Prolene. I'm not sure if we have it on this slide. Yes, here it is. So Prolene actually forms a ring with its own backbone. And this has drastic consequences for the phi and psi angles that it assumes. The phi and psi angles are torsion angles across the three atoms of the backbone. Basically, they measure in what orientation a backbone is rotating. We have a lot more on this in the course Structural Bioinformatics in period 4. And then finally there's glycine, which is unique in that it doesn't have a residue. It only has a hydrogen atom, where other amino acids have these bigger chains. And this too means that it's much more flexible than other amino acids, which also means that it has different psi and psi angles than you would expect from normal ones. Okay. So we briefly mentioned hydrogen bonds. Do I want to go into this before the break? How are we doing? Do we need to stretch our legs already? Or can we talk briefly about hydrogen bonds? Show of hands who needs a break? Alright. Sorry man, there's not enough. Alright, 5 minutes. Okay, so a hydrogen bond is an attractive interaction between two dipoles. And this happens when an electronegative atom is in the proximity of an electropositive one, and this is roughly what that looks like. So here we have a water molecule, 2 H's and an oxygen, and an ammonia molecule which has a negatively charged nitrogen atom. So these two forces, or rather these two electric charges, attract each other. So if they find each other in solution or in an environment, they will form a hydrogen bond. This is called a hydrogen bond because water has a lot of these things, and of course hydrogen is always involved in it. Okay, so H bonds in the peptide backbone. We've shown you the repeating pattern of the atoms of an amino acid's backbone. These things also form hydrogen bonds. The oxygen of this carboxyl group is negatively charged, and the hydrogen sticking off the nitrogen is positively charged. So if these two are close, they want to stick together. Right, so these are the two people that proposed the secondary structures alpha helix and beta sheets. That's the primary structural motifs in protein backbones. And this is roughly what this looks like on an atomistic level. So if you have your backbone of amino acids and you align these all in the cis rotation, then roughly every 3 amino acids your backbone has made a full turn. In doing so, the hydrogen bond acceptors of the one atom are within range of the donors of the other. So you have this stack of small hydrogen bonds stabilizing this helix structure. Let's see, can I show anything else? Right, so this is on a scale of roughly half a nanometer per turn, very, very tiny. And the oxygen and the nitrogen ones are important here. If you don't have a cis alignment, but rather the rotation is transverse, then you can end up with a beta strand if 2 of these chains lie side by side. So here again, you have 1 strand of backbone, here you have another, and here you have the third. So this is 3 lines of amino acid backbone that form these stabilizing hydrogen bonds between them. And if we render this in a cartoon, it looks something like this. Right, so you're either side by side or you're face to face, and the hydrogen bonds between them make so that this structure is actually fairly stable. Hydrogen bonds in a beta sheet are formed with the neighboring strand. The strands may either be parallel or anti-parallel. So you can have strands that go like this, or you can have strands that go like this, and they will still form hydrogen bonds between them. Question?
Yeah, so how can you tell what is like the direction of these like product types?
So we've shown this. No, this. A protein is encoded from its N to its C terminus. That this C will latch on to the next N, and suppose this is now a 2 amino acid peptide. This would be its N terminus, and this would be its C terminus. So the direction of this peptide would be like so. Clear?
Yep.
Anyone else? Okay. Then I propose that while you ruminate on hydrogen bonds and how they form these atomistic structures that give rise to secondary structure, stretch your legs, grab some coffee, and see you back here in 15.